All right, today we're doing something a little different. We're going to unlock the doors and take you inside a real biochemistry lab. We're talking a full tour of all the essential tools that, power actual scientific discovery. Everything from the simplest piece of glass to some seriously complex machines. So let's get right into it. So when you picture a scientist, what comes to mind? Probably someone in a white lab coat, right? Maybe staring really intently into a microscope. And yeah, that's definitely part of it. But honestly, a huge chunk of their day is spent using this incredibly specialized toolkit to get samples ready, to separate them, and to figure out what's inside.
And that really gets to a fundamental truth in science, right? Having a great question is awesome, but it's only half the battle. To get the answer, you've got to have the right tools. So for our tour, let's start at the very beginning with the absolute basics.
Okay, before we get to any of the flashy, high-tech stuff with all the blinking lights, we have to talk about the foundation. I mean, the thing that almost everything else in the lab depends on. And that's the glassware.
Now, this is not the same glass that's in your windows or your drinking cups. Not even close. This is borosilicate glass, and it is pretty much the unsung hero of the lab. See, its unique chemical makeup, mostly silica and boric oxide, means it can handle crazy temperature changes without cracking. You can heat it up, cool it down, and it's fine. Plus, it doesn't react with most of the chemicals you'd ever use. It's tough, it's reliable, and you can see through it. It's pretty much perfect for experiments.
And what's really cool is how this one special material gets shaped into a whole family of different tools, where every single one has a very specific job to do. You can kind of think of it like a chef's kitchen, but instead of food, you're working with molecules.
So first up, you've got the general purpose crew. The stuff you see everywhere. Test tubes. Perfect for those quick visual experiments, like seeing if something changes color. That's what we call a qualitative experiment. Then you have beakers. These are the workhorses of the lab. Basically, they're all-purpose mixing bowls and cups. And flasks, well, they're your go-to for mixing up and storing your chemical solutions, or as scientists call them, reagents.
But get this, even flasks get specialized. You see that conical one? The one that looks kind of like a pyramid? That's perfect for swirling liquids without them splashing all over the place, which is super important for a process called titration. Then there's the volumetric flask, with that long skinny neck and that one single line on it. Its job is to measure one and only one super precise volume. And that round-bottomed one? Well, that shape is all about making sure liquids heat up nice and evenly. Every shape has a reason.
Okay, moving on to the measurement masters. Measuring cylinders are great for getting a pretty good idea of a volume, when close enough is good enough. Funnels? Well, they're simple, but you'd be lost without them for pouring stuff without making a huge mess. And then there are pipettes. Pipettes are a whole different level. They are all about delivering an exact, tiny, repeatable amount of liquid every single time. And that is absolutely crucial if you want your experiments to work.
But science doesn't just stand still. Glass is the classic, for sure, but modern labs have also brought in plastic alternatives and some pretty serious tech upgrades to these basic tools.
And the pipette is the perfect example of this evolution. I mean, we went from these traditional glass tubes where, and I'm not kidding, scientists sometimes had to use their mouths to suck the liquid up, to these modern auto pipettes. Talk about an upgrade. Now you just set the volume, push a button, and you get a super precise amount instantly. It's faster, way more accurate, and a whole lot safer. A total game changer.
And sometimes even that super tough borosilicate glass isn't enough. There are some really gnarly concentrated acids and alkalis that can actually eat away at glass over time. For those, you turn to plasticware. It's often made of polyethylene, it's pretty much unbreakable, and it can handle those harsh chemicals no problem. It really fills a critical niche in the lab.
All right, so we've covered all the hand tools. Now it's time to check out the heavy machinery. We're talking about the bigger instruments that really define the power and capability of a modern lab.
And first, we're going to look at the tools of separation and disruption. These are the machines whose entire job is to take things apart, to break open cells or pull molecules away from each other, so you can see what's really going on inside.
Say hello to the centrifuge. The easiest way to think about it, it's like a salad spinner on steroids. I mean, one that spins thousands, sometimes hundreds of thousands of times a minute. That incredible spinning force separates things by density. It pulls the heavier, denser stuff down to the bottom of the tube. This is how you separate plasma from blood for a blood test, or even pull out specific little organs, the organelles, from inside a cell.
And of course, there isn't just one type of centrifuge. You need the right tool for the job.
You've got your standard clinical ones for routine stuff like blood work. But what if your sample is sensitive to heat? Then you need a refrigerated one to keep things cool. Need to separate really tiny parts of a cell? You'll need a high-speed model. And if you want to separate individual macromolecules, like proteins or viruses, you bring out the big gun, the ultracentrifuge. But with all that spinning power comes a huge responsibility. Seriously, if you put an unbalanced load in one of these things, it can literally tear itself apart at high speed. It's incredibly dangerous. So rule number 1, always, is to perfectly balance the tubes. And of course, you never, ever open the lid while it's spinning. Safety is absolutely everything with these machines.
Okay, so you've separated your sample. What's next? Well, now you've got to measure and analyze what you've got. This is the moment of truth. This is where the data comes from, where the answers to your big questions finally start to appear.
And just like with everything else, there's a specific instrument for every kind of question you want to ask. Need to know how acidic or basic a solution is? That's a job for the pH meter. Want to know exactly how much of a certain biomolecule is in your sample? The spectrophotometer does that by seeing how much light the sample absorbs. And how about finding out the levels of metals like sodium or potassium? A flame photometer can do that just by analyzing the color of a flame. It's pretty amazing.
And just like we saw with the pipettes, analysis has gone high-tech in a big way. Enter the autoanalyzer. This machine is a complete game changer. It can run dozens of different tests on one single blood sample, all at the same time, all automatically. It's checking your liver function, your hormone levels, cholesterol, you name it. Ever wonder what happens after you get your blood drawn at the doctor's office? This machine is usually the answer. It's the engine of modern medicine.
But here's the thing. None of this works. Not the fancy machines, not the simple glassware, if your stuff isn't perfectly, spotlessly clean. One tiny, invisible speck of contamination can throw off an entire experiment and waste weeks of work. That's why cleaning in a lab isn't just a chore, it's a science all on its own.
Yeah, it's way more complicated than just using soap and water. Did you know that brand new glassware actually needs to be soaked in a weak acid solution overnight before you can use it? For just routine washing, detergent is fine. But if any glassware has touched clinical samples like blood, it has to be completely decontaminated, usually by soaking it in a bleach solution. You take zero chances with that stuff. And once everything is clean, that whole cycle, prepare, separate, analyze, and then clean again, it just starts right back up. And this is happening in labs all over the world, 24 hours a day, 7 days a week.
So, there you have it. From a humble little glass test tube all the way up to a fully automated analyzer, every single piece of equipment has its part to play. These are the tools that let scientists take a simple question, a moment of curiosity, and turn it into real, hard data. They're what turns what if into we know.
And these tools, they're used to tackle the biggest challenges we face. We're talking about curing diseases, understanding the very building blocks of life, and inventing the future. So I'll leave you with this question. If you had access to this entire amazing toolkit, what's the one great challenge that you would solve?